speaker, Dr. Chandra Varma, for the talk.
Good morning once again. I know that Dr. Chandra Varma does not need any real introduction to those people who are already here and those who have come earlier. But for the sake of this session, I would like to introduce Dr. Varma as an alumni of IIT Kanpur, and then he moved on to York University in UK to do his PhD. From then thereon, he moved on to Singapore, the A-Star Institute of Bioinformatics, where he is doing work on biomembranes, structure of proteins, and how they are related to biological functions. Yesterday we had Sir Blundell talking on membranes and then drugs. So any drug that we use has to cross the biological membrane. And we assume that they either interact with the receptor or they have to somehow structurally integrate with the membrane, get inside the cell, and then even then it may have to cross the nuclear membrane, again another membrane barrier, and maybe it will be directly acting on the DNA. DNA, as you know, is an inert molecule. It does not partake in any reaction. I repeat, DNA does not partake in any reaction. It stays silently in the nucleus, and then how it works is through proteins. Okay, so it sends message to make a protein, and the protein a workhorse which will facilitate all that happens within the cell. Now here's a man who is working on that proteins which are workhorses. So any structural difference in the protein will show itself in the whole system that is working within the cell. So we all scientists, many of us are working at least directly or indirectly with drugs, hoping that we'll have a drug that will be acting on a particular disease, making it cheaper or making it very effective. Maybe we do get a few drugs that do act on it, but within a few years you find that there is some sort of a resistance against it. And then we get depressed, but of course the happy part is that keeps a scientist in jobs. Okay, so we keep on searching for drugs. There is a new drug, there is a resistance, you go for another search, you keep on working. So that keeps us employed. But anyway, there is always a constant fight. So Dr. Chandra Varma will be talking on membranes, developing novel therapeutics, and then I'm surprised that he's talking on staple diets. I wonder how that is going to help us in understanding drug resistance and how drugs will act. That is perhaps what Ayurveda says, where it says, let food be thy medicine. Okay, I believe you must have heard about that. Allopathy says, let medicine be thy food. Ayurveda says, let food be thy medicine. So there's a classic difference between that. Now he is introducing that particular subject where maybe I'm not sure what he's going to talk, but then since the title says that, he may introduce us to tell us what material in the diet can act as drugs. Dr. Sharma, as you know, as I have to introduce him, so he is working on membrane dynamics, how the protein structure is influenced by its sequence, and then how this particular structure can be influenced by other molecules, and how the structure and biological functions are related. So he has a great number of publications. There's a long list in the net. There are a few which has been highlighted here. He has been working on MRSA, which, you know, that particular results came out from All Institute of Medical Sciences, where India was blamed for producing such type of bacteria. So it's working. So he will be talking on, his publications include such exciting work, and then amino acid modified xanthone derivatives, which are all exciting fields. So let's hear from his mouth what he has to say about this particular topic. I'm supposed to welcome you also, so that's part of the welcome. I'm sorry that I missed out, so welcome to this race.
Well, good morning, and thank you very much for the kind invitation. I'm an antique here, really. I've been here, I think, since Pavan conned me into coming here in the first Bioindica'. In any case, it's great to be back here. I was telling Achu that, we planted trees. I bet none of you know this. Well, apart from Pavan. And they've grown away. It's amazing. The surroundings are just absolutely fantastic. Okay. So, since he mentioned Ayurveda and food, I came across a quotation yesterday, an Egyptian or a Greek quotation, which partly applies to me. It says that of the food we eat, a quarter is what we need to live, and the other 3/4 is for the doctor to stay alive. So, anyway, so Tom yesterday, of course, for those of you who attended his brilliant lecture, laid out the field, right, in specifics. So where the important areas are to focus on. There are areas that need immediate and urgent attention, and one of them, of course, as Tom highlighted, was about resistance, in which, of course, they are doing an amazing amount of work. So this will go into some depth in one small subset of Tom's elaborate talk. Okay. So why does resistance arise? Yeah. Apart from being perhaps an innate property of any organism to survive against external perturbation, let's just look at what exists in the current therapeutic world. That when you go to a doctor, there are two major classes of therapies, right? But before that, you ask this question that these three proteins or these three pathways, for those of you who are biologists or cancer researchers, the MYC, RAS, and P53 are extensively well researched. Yeah? There's beautiful amounts of biological data, genetic data, whatever, right? Yet, with all this data, none of them have a working drug in the clinic. Yeah? It's been shown that they are very important for cancer. Yet there's nothing in the clinic. So why might that be? So Tom has referred to this problem earlier. I'll elaborate on it a little bit using an example. So here's an example for P53, right? You see that P53 in it's a small subset of cancers is very highly mutated in these cancers. And so, P53 is an important protein to maintain the health of the cell. And if things go wrong, then the cell, clearly starts behaving aberrantly. So the reason is, the following, or maybe the following, that the two classes of therapies we have are small molecules, like the xanthone derivatives he mentioned, and the protein therapeutics. So if you look at the small molecules, they're all about 100 atoms. They're quite hydrophobic. These are the ones that enable them to go across membranes, to go inside the cell very easily, right? And they are usually found in the active sites of proteins bound to these little caves, okay? And that becomes significant in our ability to design better small molecules, okay? We'll see that in a second. In contrast, you have protein therapeutic. So Herceptin for breast cancer, for example, is a good example. They are large molecules, thousands of atoms. And because they are large, it's not easy to get them inside cells. And I use the word it's not easy because there are some instances of large molecules getting inside cells. But nevertheless, these antibodies do not get inside cells, as is commonly thought. And so they only act on extracellular receptors quite effectively, yeah? And they're very specific. But they make large contacts over their extracellular receptors. What do I mean by that? So you look at this picture, and what you see over here is a kinase, which is the target of a very successful anti-cancer drug called Glivec. Okay, it's been a lifesaver. It's one of those rare miracle drugs that has saved thousands of lives. And here what you see in yellow is the active site of the protein, kinase, and in blue, where you can barely see, is that small molecule Glivec bound to it. Okay, so it goes inside, it binds there. So if you're a good chemist, what do? You look at how it interacts, and then you can add little functional groups to improve its affinity for that particular target, which is what commonly you do in drug design. In contrast, what you see in green here is Herceptin bound to the extracellular domain of the HER2, an EGFR family receptor. And you can clearly see that it makes a lot of contacts over the surface. What's the problem here? The problem is that unlike here, where you have a few number of contacts, which you can easily chisel, you play Lego with them, you can say, okay, here I add this, here I add this. Here it's a nightmare, because where do you add what to improve the affinity? Okay, there are techniques like alanine scan and all, which have been quite useful in making better antibodies, but nevertheless, it's a complex problem. It's not that easy, okay? So you have these two extremes, right? These are bloody expensive ones, okay? These guys are not expensive, but they have a problem. And the problem is the lack of specificity. Yeah? So this active site, for example, in the kinase, evolved to bind ATP. It did not evolve to bind Gleevec. So when you attack it with Gleevec, it's going to do its best to throw Gleevec out and rebind ATP, and that's the basis of resistance. As Tom mentioned yesterday, that they will throw your interrupting molecule and rebind the substrate. In this case, the substrate is ATP. Okay. These guys have a lot more specificity. That's why you do not see so much incidence of single point mutations that disable the binding of these. You have other nightmarish pathways, crosstalks and things like that come up as part of resistance, upstream and downstream signaling. So what does this mean for therapy? If you do a simple calculation, and Tom alluded to it yesterday as well, actually with the current therapies, we are only covering 10% of the proteome. That's it, 10, or maybe if you are charitable, 20%. So the whole 80% of the human proteome, which is involved in all these complex signaling pathways, that is waiting to be drugged. Okay, so how do we do it? Why haven't we done it? That's the question. And the question is here. Most of the intracellular protein targets are part of protein-protein interaction, signaling pathways, this, that, and the other. Okay, and the problem with them is this. You see, this is a cross-section of two proteins interacting with their protein partners over here. And what do you see? You see a lack of those little holes, these caves or cavities that will allow you to have a small molecule that binds there, and then you can, chisel it away to make a good molecule. Yeah, that's the problem with these. These are large, flat, interacting surfaces. What do you do with it? And once again, for those of you who attended yesterday, Tom very nicely showed very beautiful examples of these kinds of surfaces. Okay, so what can we do about it? Yeah, what lessons can nature provide us to try and tackle this problem? So here we should thank the team. And okay, I don't have 37 languages, but I counted today we have 16 languages. A lot of them are Indian, of course, but nevertheless. Okay, so let's take P53 an example and see what we can try and do about this problem. Okay, so here is P53, a small cross-section of the P53 pathway. The important thing to remember is that P53 is likened to be the guardian angel of the cell. So anytime there is any stress in onto the cell, so whether it's oncogene activation or whether there is DNA damage from radiation, or you might have a chemotherapeutic drug, P53 becomes active and then it senses DNA damage. So it doesn't want the DNA damage to be sent out to the daughter cells. Yeah? So it has to stop the cell from dividing and do an error check. That's what P53 does. Cell cycle arrest happens, there's an error check. Can this damage be repaired? If it can be repaired, it is repaired and the cell starts dividing. If it cannot be repaired, the cell has to die. Yeah, so for example, well, Tom is quite used to traveling, but if you see your Caucasian colleagues, you see they spend too much time in the sun and their skin starts peeling off, and that's it. That's P53 in action, removing those skin cells from becoming oncogenic because of too much radiation. Okay, that's why that's apoptosis. So P53, of course, is very important, so it has to be constantly kept in check. It's a constitutively active protein. It's constantly being made and removed until there is a signal that we need P53. And one of the proteins that does it is called MDM2. Okay, so that's the important thing to remember. And so what happens is that in a lot of cancers where P53 is not mutated, MDM2 is overexpressed. What does that mean? Why is it cancerous? Is because even if the cell is sensing a cancerous change and it is trying to tell P53, come on, do something, there's so much MDM2, it doesn't allow P53 to do anything. Okay, so here is an opportunity. If we can understand how P53 is held by MDM2, then maybe we can disrupt that interaction using a small molecule or something else, right? That's the idea of making this therapy. And so to do that, you look at the structure, of course, so many thanks to the people who started this whole field, which is Dorothy Hodgkins and Tom Blundell and so on and so on. So this is the structure of P53. What you see here is a helix. It's a small part of P53 bound to a part of MDM2 over here, which is in the surface. Okay. Now this helix has to be a helix. Why? Because a helical motif is one of the best secondary structural motifs that can present a hydrophobic surface on one side and that can present a water-soluble surface on the other side. Okay, and it is quite robust in itself as a secondary structural element. These things have become very important when you start trying to interrogate protein structure. Okay, so now we have to do something. We have to ask, what are the specific interactions that P53 makes with MDM2? Why? Because you want to make a molecule that will mimic those interactions. Why? Because you want to inhibit this pocket. You do not want this pocket to bind P53, you want P53 to be free in those cancers. So you try and understand what are these interactions, and you make a small molecule that will do better than those interactions. This interaction is conserved at least over 1 billion years. Okay? So when they did a cell-based screen, they found this. Okay? This molecule called nuclein. Okay? So this is how it is. You can see over here when you superpose the two, that nuclein mimics those interactions, and due to various thermodynamic issues, it just does a much better job. Okay, so what did people do? People started making all sorts of variants of that molecule nuclein, and currently I think there are 9 companies that are desperately trying to get a molecule into the clinic, and I think 2 have already stopped because of extreme toxicity. Okay, so fine. So what's the problem? The problem is this. Look at these 2 pictures. Okay, this is a sphere representation of that small molecule nuclein. This is a sphere representation of the P53 peptide that I showed you. Okay, there are 2 crystal structures. Look at, focus over here. Okay, look at this. There is no yellow patch here. Anyway, there's a methionine residue here. If you mutate that methionine to an alanine, what has happened? In both cases, you have lost some amount of contact. The difference between these 2 is that this occupies or makes very, very few contacts with MDM2. So if you're losing a critical contact here, it has lost a lot of free energy, and so it gets kicked out. Okay? Whereas P53 being a peptide has a lot of other interactions. It's like Mr. Gulliver. It's those weak interactions Tom was talking about. Lots of weak interactions, and yet there's a lot of robustness in that interaction. So if you remove this, P53 just readjusts there. It's a very promiscuous peptide. It readjusts over there. It's like on a dancing floor, your irritating partner, you've had a fight and he or she will go off with somebody else. It's exactly like that. They don't care. It'll just make another stabilizing interaction. And this is the basis of resistance for small molecules. You see a small change, well, it's not a small change, nevertheless, a change of losing a key contact with a small molecule here renders it ineffective. Yeah? But because it doesn't happen here, does this provide us a clue that maybe we should look at this intermediate space between the large molecule of antibody and the small molecules that we looked at called peptides? What can peptides offer us? We can see. Okay. So just to give you a relevant example of this in the clinic, look at this picture. Okay. So this is another view of that same Gleevec over here, bound to that BCR-ABL kinase, and focus on this. Okay. Invariably, all patients on Gleevec, not all patients, but most of them, Gleevec becomes ineffective because of this change. Watch. Okay, so now it's making a nice hydrogen bond, this 3-ene with this nitrogen here, oxygen, there. Yeah, this 3-ene mutates into an isoleucine for various reasons. Yeah, and so therefore you can see this clash, and so the drug becomes ineffective. So then people have made 2nd generation drugs. When they become ineffective, they make 3rd generation drugs, and keeps people in job, as you rightly said. Okay. So there's a problem with small molecule therapy, clearly, right? And large molecules, we want to stay away from them because they're very expensive. As Tom rightly highlighted towards the end of his talk yesterday, there's an urgent need to make something small, yeah, and cheap and easily synthesizable and easily available to everyone. So if you look at the experimental data on P53, what can peptide studies tell us? Look at this graph, right? Look at this table. This was the original sequence of P53 bound to MDM2, and when they did phage display and optimize the peptide, this is what you get. Such a huge change in affinity from a bad 8,700, the micromolar to a nanomolar affinity. So that's a good change. So that tells you that if you study the interactions between the peptide and its target and try and understand what those interactions are and try and make reasonable logical changes to them, you can actually gain huge amounts of affinity. Okay? So then what's the problem? The problem is that if you have a peptide, imagine I inject a peptide into my bloodstream, what will happen? It'll get chopped up, right? Because the peptide is always flexible, there are proteases waiting to chop up these kinds of things. So what do? So people have been trying to stabilize peptides using all sorts of funky chemistry, which has rendered very, very nice peptides, very stable peptides, except for the one critical thing. They do not go inside cells. And that is, of course, then it's useless. Because if something doesn't go inside the cell, then, So what do? So in this process, somebody decided to exploit this particular technique, which actually was, a very old technique. But nevertheless, it has been taken on more recently by some people in the US. And it seems to be quite a useful technique. And the technique is the following. Say here is my target and I want to make a peptide, but I want to make a stable peptide. Yeah, what do? Here is a protein-protein interaction, right? You see this, and this is part of another protein. So I chop up the interfacial peptide, okay, of this pair. I chop off this bit. Okay, so I have two proteins interacting. One of them has an alpha helix at the interface. I cut out that alpha helix and see if it is a competitive inhibitor, which I find it is. Because the specificity is exactly the same that nature provided. I haven't done anything yet to it. But now I would like to stabilize this as a helix, which I know it doesn't exist because it's flopping around in solution. Yeah? Because if it's already a helix, not only does it prevent it from being chopped up by proteases, it also enables it to bind much tighter for thermodynamic reasons of entropy. So what you do is you figure out which 2 points in this peptide you will introduce this amino acid. You see these amino acids here, it's got a methyl group here and a polycarbon chain here. And then with a one-step reaction, you can make this. You can just click it into place. Okay, so now you have a helix and you've got this staple, just like you're stapling. You've got a staple here, and this is what happens. So now what happens? Now you get this amazing thing. You get a peptide that has been stabilized against proteolysis. It's been stabilized as a helix. But the most amazing thing which has yet to be understood as to why is most of them go inside cells unaided. And that's critical. Because a lot of people are trying to develop peptides and proteins as therapy by adding on these cell-permeable peptides. Yeah? But cell-permeable peptides bring with them their own added confusion. Like what happens to the, is the cell-permeable peptide that you are adding onto it interacting with the surface? Is it changing the affinity? Is it going to bind onto something else and give you a side effect, right? So for some unknown reason, most of it people think is just endocytotic, is that these peptides seem to go inside cells. Okay, so this is promising, right? So you know that now you have an ability to make possibly this particular specific molecule, which is not a small molecule. It has a specificity almost of an antibody, not as much. Nevertheless, it has a good specificity. It is stable against proteolysis and it goes inside cells. Okay, so where do we go from here? So let's test this. Okay, so once in a while somebody will publish this kind of great data, right? So they've varied the staple, they've varied amino acids, and they've got a great amount of data. Some of it are really lousy binders and some of it are great binders. So for computational analysis, it's great. So you go and do the analysis and you find that in that particular case, in certain positions, if the peptide is not appropriately placed, you see what happens. It's messed it up, right? The helix is distorted. This hydrophobic carbon is pointing into solvent, which they don't like because they're hydrophobic. Whereas all the nice binding ones, the staple has stabilized the helix, and the staple occasionally is like a garland, right? So here you put the staple, it's like a garland that it goes and interacts with the surface of the protein. So you may ask, okay, but it's hydrophobic, but the protein doesn't have so many hydrophobic surfaces, right? Actually it does, and we'll come to how do you explore that in a second, yeah? But the protein is always dynamic. Right? So occasionally little tiny blips of hydrophobic surfaces become exposed. Their residence time is not long because they have nothing to stabilize them, right? So hydrophobic cavity opens, they see only water and they quickly close their eyes, right? So it's like what we do in the face of danger, quickly close your eyes, you don't want to see danger. Okay. So of course when you do that, yeah, so here you see this was the model complex, and 2 years later the group took this data and crystallized it, and you can see the resemblance is quite good. And actually on second thoughts you realize it's not a big deal. Yeah? There's no rocket science here. It's quite easy to do this. The advantage is once you have this assembly, you can make your favorite mutations which will give you that higher affinity, and so at the end of it you get an absolutely amazing binder. Look at this graph, okay, for those of you biologists. When you add nutlin, yeah, this is nutlin. This is the activation of p53. Look at nutlin. It works till here and then the cells die. And they are dying because of an off-target effect, which I said is inevitable. You cannot do anything about off-target effect because the proteins have not evolved to bind nutlin. There are enough cavities the size of nutlin in the proteome that will invariably bind your small molecule. Whereas these fellows have been designed based on the specificity nature provided. There is no off-target effects, and you can clearly see that the two peptides that continue to give p53 activation in a much better manner than nutlin. And okay, so now you see, yeah. So now you can expand your target space immensely because you have this ability now to suddenly target these protein interactions that Tom was alluding to which form almost 80 to 90% of the proteome, and there's a huge variety of targets to be looked at. And here's an example of some data from the PDB of these various staple peptides bound to various targets from various groups. Some of them we have also done. And so you can clearly see, and the list is growing actually almost on a weekly basis, because I think this is a very interesting area to tap into. So now, we have this, right? Can we do just that little better to get that additional affinity? Because you have to sniff around, right? You have to do something to make it just that little bit more effective. Yeah? So this is where we come back to this question of asking that if there are hydrophobic interactions, yeah, if I want to tap into those, because if you have a polar interaction, let's say on the surface of a cell you have an aspartic acid and an arginine, right? Your peptide has an aspartic acid and you have an arginine in the protein, and you make that mutation and you get an additional electrostatic interaction. The problem with that is that these guys are exposed to solvent, okay? And each polar amino acid prefers to have lots of water swirling around it. So just because you make an ion pair doesn't mean it's going to go and form an ion pair, because they would rather be like this because of various entropic advantages, etc. So then shall we tap into the hydrophobic pockets? But where is the hydrophobic pocket? I can't see it in the crystal structure because most structures have been crystallized in water solvent environment, right? But we know that they must exist because the protein is fluctuating. How do I capture that? Okay. So when you do molecular simulations to understand the dynamics of any protein or biomolecule, you take your system and you surround it with water molecules normally, right? And then you watch how it dances around. So why not infuse in this solvent of water small benzene molecules as an effective probe? Let's ask what happens. So I replace some of these water molecules with a hydrophobic called benzene and see what happens. And this is what happens. Now you see the benzene swirl around, okay? These are flies around, whatever. So, and then you begin to see little hydrophobic pockets open up, and then the benzenes go and concentrate over there. So suddenly you are exposing these little pockets, you are stabilizing those hydrophobic pockets, because as the pocket opens up, it doesn't see water anymore. It sees something it likes, because the hydrophobic benzene is suddenly there in front of it, so it grabs onto that. Okay. So watch this. This is an even better representation of that, because this loop binds to this hydrophobic cavity of MDM2, and you can clearly see that the benzene is even able to displace this loop. This we never see in our long MD simulations either. You can do very long simulations, we very, very rarely capture this, because this loop tries to lie over this active site, which is hydrophobic. Okay, but here you can see that with just a simple little protocol, you can actually start opening up very, very new druggable cavities in proteins. Okay, so what do you do? How do you test it? So that PhD student, he had done his PhD half between me and half between Tom's department, and as part of his PhD he had done this work. So you can clearly see that they exposed a hydrophobic pocket here, added this moiety over here, and there's a crystal structure of what he had predicted. And similarly, So when we are talking about this work, these people, Martin Noble, he agreed to try and crystallize it because they've developed an effective crystallization method for MDM2, and this is what you get. So this was the staple peptide originally designed against MDM2. Here you see the staple, and then when we were sniffing around with this benzene, it opened up a new pocket here, and we put a phi here, and suddenly you had a huge affinity change. I think it went from about I think it's almost close to picomolar. Not exactly there yet, but almost coming to picomolar affinities, just by the discovery of this pocket and the addition of this phenylalanine group over here. And this is a crystal structure, by the way, okay? So you can clearly see that this seems to be a very, very powerful new technique to be able to do. So what does this have to do with resistance? Yeah? So let's come back to resistance. So we had some hints, right, that a peptide is going to be this promiscuous dynamic peptide which will avoid resistance appearing, just because it will not avoid resistance appearing. Even if resistance appears, or maybe appears is not the right word, maybe it already exists in the pool of different mutations, I don't know. Nevertheless, if it encounters a resistant mutation, at least at a single point level, then clearly peptides have something to offer by way of avoiding the lack of inhibition of those mutations. So can we do something with staple peptides? And so to understand that, there's one other thing that I need to focus on and then we'll finish. How much time do I have left? 2 minutes? 5 minutes? I have half an hour. I don't have that much to talk. Sorry. Okay, good. Nice for the next speaker. Okay. So now you remember that there is one aspect that, you have this molecule binding there. Yeah. The molecule binds here. This amino acid changes. It becomes a larger amino acid and it kicks out my molecule. Correct? That's a very common form of single point mutant resistances. What's the other point of resistance? If we can understand the various forms of resistance at a molecular level that can arise and that we can do something about, then we will take this technology over there. Yeah, there's some purpose to that. So let's just look at this example and ask. When P53 binds MDM2, normally, what is the biological signal that allows P53 to escape? It's all right to put in a small molecule, right? But it's happening all the time. As I'm speaking, probably, there's some stress and, my P53 is being active. So what is the biological signal? Of course, one of them is phosphorylation. It's very well known. In most protein-protein interactions, there's phosphorylation, and there are various other post-translational modifications. Let's look at phosphorylation. Okay. So it is known for sure from experiments that this threonine, this is P53, this threonine, when phosphorylated, allows for the alleviation of this interaction. How does it happen? Let's see if we can understand this. So this is a side view of the protein MDM2 in shade, and this is P53 helix as we saw. So now, here is the threonine. Okay, I don't know if you can see at the back, but the threonine lies here. What is stabilizing this as a helix? Yeah, because I told you that this thing is flopping around. What is thought to, now you have to refer back to Tom's talk yesterday about, a little bit of binding and then there's a folding over there. So in the ensemble of these P53 peptides, you have some which are helical. Okay? So some of them are trapped, small helices, so they come here, they are attracted to this due to some electrostatic steering mechanisms. And then once they are bound there, then the groove allows that thing to fold into a helix in a manner. Okay? I mean, this is not the place to detail that interaction, but it's been well characterized. So what's going on here? So here, that helical pet is stabilized by this threonine making a hydrogen bond with aspartic acid which is next to it. Okay? Aspartic acid, negative charge. So now, you bring in a negative charge on that threonine because phosphate is a negative charge. What do you expect? You expect a huge repulsion between the two. Okay? And because of that repulsion, this helix will not form. That was the intuitive understanding, and that makes sense, that the helix won't form, so P53 will not bind, and that is how phosphorylation allows P53 to be free and do what it has to do. Is that really right? So when you do this, at least when you test this through simulations, you find that is not right. Okay? The helix doesn't change no matter what you do. This pinkiness tells you it's a helix. Why? Because of that bloody promiscuity of the peptides. Yeah? The side chains are very promiscuous buggers. They change partners. They don't care. You don't have to break the helical backbone assembly, helical backbone structure for a side chain to just rearrange and form new interactions. So you can phosphorylate it as much as you want. You'll just go and find another hydrogen bonding partner. That's all a side chain needs. It's just, these side chains at least, they just want a hydrogen bonding partner. Whether it is water or whether it is something else that is more stable, they don't care. If you're a graduate student, or if you're a boss of the graduate student. But then, you may ask that now we are looking at two things coming together. This one of them is suddenly carrying a big negative charge, right? So maybe it just has something to do with very simple physics of the attraction of two particles. Might it have to do with that? So you ask. And there are very simple methods on the web to create these kinds of electrostatic surfaces of proteins. So here is MDM2. This is all blue, meaning that MDM2 is largely positively charged. But you see little islands of red. There is some negative charge. We know P53 has to bind like this. Yeah? The crystal structures tell us, lots of experiment, mutagenesis tells us it has to bind like this. If it has to bind like this, what happens? If there is a phosphate group here, then the phosphate will lie directly in line with that negative charge, right? See, the threonine lies right above the negative charge. The threonine has to be there because the geometry of this constrains it to be there. It cannot go anywhere else for optimal binding. Because the threonine is there, if it was carrying this phosphate group as a negative charge, it will come there, but suddenly it is kicked out because there is a negative charge here already. So it comes and then comes and then. In kinetic terms, this means that the rate of coming off should increase quite tremendously, right? So that's okay. So here's a close-up, right? So here you can see the phosphate lies right above that offending negative charge. Okay, and so Alan first was doing these kinetic experiments, and you can see over here that when you have unphosphorylated p53, there is your K off at about 2. When you have phosphorylated a threonine 18, it goes up tenfold to about 20. Okay, so there is some margin over here that maybe that's what's happening. How do we test it? You find out what those negative charges are arising from, what amino acids cause those, and you mutate them out. Yeah, that's what I'll go and tell my experimentalist friend. Look, I have suggested a new MDM2 mutation where this negative charge that was preventing p53 phosphorylated from binding, I've removed it, I made it positive. So now p53 should bind. Whether it's negatively charged with the phosphate or not, it should bind because the negative charge is gone. And of course, you do the experiments, and of course, it's not rocket science that it does work like that. But then the next step you may ask is, does this happen in the cell? In a cell which is under pressure to become cancerous, to prevent p53, because see, if p53 is wild type, the cell is constantly signaling, right? Come on p53, come on p53. But MDM2 is the one that's causing the problem. There's tons of MDM2, so it's just suppressing p53. So maybe one day when somebody sequences the genomes of cancers and finds out the sequence of various MDM2 mutants, it is quite possible that you'll already find MDM2 mutants which exist like this as a resistance. Why? Because MDM2's job is not to let p53 go. It will do anything to avoid p53 from leaving. So wild type p53 or phosphorylated p53. When both are wild type, phosphorylated p53 will not bind. However, if MDM2 mutates, phosphorylated p53 will also bind there. So is this, right? So given these, once you compute these, and these are simple computations to carry out, okay? These are not very, very complex things to interrogate, and it sort of links back to this database that Tom is developing very deeply about, you know, the relationship between amino acid changes and their surroundings. That's very, very critical to get a molecular level understanding of these. And so what happens is that, oh, I've lost that slide. No, I haven't lost that slide. Sorry, I'll just go back. Ah, never mind. Don't matter. So what happens is that we turn to these people in the neighboring lab, in the neighboring institute, and ask them if there is a way of evolving MDM2 under this pressure. Okay, so there are these in vitro evolution techniques where you can take a protein of your choice and allow it to evolve under the pressure of a molecule like nuclein as opposed to in contrast to the substrate. Okay, so you'll get thousands and thousands and thousands of mutants that you generate essentially, and for each of these mutants you're essentially very rapidly testing whether they bind nuclein or whether they bind p53 or whether they bind a molecule of your choice. Okay, so he does this whole scans of resistance, and of course you will find resistant mutations like that. That's not surprising, but the nice thing is at the end of it, if you look at this table, is that you watch here. So the p53 peptides, and there's an example of 3 particular mutations that arose as a form of resistance. Look at nuclein binding. Yeah, nuclein binding has gone up worse and worse and worse. That means these are the kinds of mutations of MDM2 that do not like molecules like nuclein and will throw them out, and yet rebind p53 peptide. Okay, so what do? You look at this column. You can clearly see that the staple peptide seems to work absolutely effectively against all the resistance mutations. And after this, he followed it up with experiments in asking the question that will MDM with the staple peptide give rise to resistance? And so far, in this experimental setup, they are unable to yield any mutations that are resistant to the staple peptides. Why is that, you may ask? Because now you have this advantage. As I said here, look at this mutation, okay? It's gone, right? Whereas a peptide exists over there despite that mutation. I've showed you already. Well, the staple peptide is nothing but that peptide, except that we have made sure that the staple enables that peptide to bind as that peptide. So you have all the specificity of the p53, which has naturally evolved over billions of years. So you have that specificity. In addition, you have the specificity of that peptide being very stable. Remember, it has to be a helix. So now you have clammed it into a helical position. So you don't have to worry about, oh, look, all these peptide molecules are floating around in different conformations. Only a few of them will really bind very well to this, and so affinity will be gone. Nothing. All you have in your solution are these dynamically varying, but within a constraint, these staple peptides. Yeah. So you have that advantage, and the best thing is that, of course, because of their promiscuity, their ability to readjust very easily, they do not give rise to resistance. Okay. So it suggests that this, in summary, you have this particular tool. Okay. It might be a new class of medicines. That remains to be seen. Of course, there's a long route to getting them out as a medicine. But the interesting thing is the following. As an experimentalist, you want to study a particular pathway, right? There are these protein-protein interactions that make this pathway. Wouldn't it be nice that instead of putting small molecules into the cell, which will disrupt your interaction, it will also disrupt some 10 other interactions? Wouldn't it be nice to have a small, nice, specific reagent for each protein-protein interaction? Of course, it will be nice to have one reagent that reads out everything, but of course, that's crazy, right? So for each of your particular protein-protein interactions, it will be just very nice to have a specific reagent which you are reasonably sure will not have an off-target effect. So all the biological readouts you see, you can be very sure that they are as a result of perturbing that particular pathway. Okay, so I think I shall stop here now. Thank you very much.
Thank you, sir, for that inspiring talk about the importance of protein-protein interactions and staple peptide. Now the session is open for discussion and questions.
Good.
Chandra, it was fantastic.
Thank you.
As usual. I have one point to check with you. In here, the design of the peptide, the inhibitor of protein-protein interaction, in this case the MDM and P53, it is designed on the basis, with enormous learning that comes from the crystal structure of the P53 peptide and the MDM protein complex.
Yeah.
Now, if I don't know whether there is a crystal structure available between P53 and MDM, and if there is one, I would imagine, I could be wrong, like we note in many protein-protein complex crystal structures. If you look at what are the what is the region in one protein that binds to another protein that comes in pieces.
Yes. Yeah.
Now we want to compete with P53, not P53 peptide.
Yeah.
So are you facing any disadvantage in not able to
Right.
use the crystal structure?
Excellent. Yeah. Excellent. Yeah. So essentially, what he is trying to say is that the whole story, even between P53 and MDM2, is not as simple. Yeah. This is just a small snapshot of a small fragment. Yeah. But the actual interaction is like this. Okay. So P53 interacts here with MDM2, as I showed you. P53 also interacts with the region over here that I didn't show you. Okay. In this case, the biological readout is telling us that it works. And there are other experiments which have nothing to do with this, which have shown that if you just disable this interaction, it is sufficient to disable MDM2 P53 interactions to lead to a biological effect. Having said that, it is also known that the second interaction that I didn't talk about is very, very important and is regulated allosterically by this fellow. Why is that important? Is because this is the interaction that enables MDM2 to do its actual function. And that function is it binds P53 here, interacts here, brings a ubiquitin molecule, attaches it to the lysines on the DNA binding domain of P53, and these lysines then, depending on the conditions required, are either a single chain of ubiquitins or a branch chain. Okay. The single chains of ubiquitins actually enhance transcription. The branch chains of ubiquitin are taken to the proteasome for degradation of P53. Okay. So there will be a complexity at some stage, because right now we are not at the level of time scales here. How long does this need to stay inhibited before this action is nullified? You see? What we also do not know is whether, so there's somebody in Edinburgh trying these experiments, whether in the presence of the staple peptide, you already see this interaction happening weakly, whether if you stabilize it for long enough, will it start ubiquitinating or not? Okay? But this we can't comment because we don't have that full model. So you're right that we are at, at a disadvantage of not having a model. But with the current information that we have and the experiments that have come out as a result of this, it is sufficient enough to suggest that just this inhibition, in this case, and if you look at all the work that has come out of this guy called Walensky in Harvard Medical School, in a range of processes, Notch pathway, Wnt, MCL, etc., it appears that the regions identified, at least from crystallographically, inhibiting those seem to have an effect. So they have shown even in mouse models of tumors that they are sufficient enough to lead to a cessation of the tumor. So it remains to be seen, but it's a very, very good point. It's a very valid point. I think a lot of people in the field are well aware of these disadvantages we have just based on crystallography. That's why you need this huge interdisciplinary effort. So this work involves, about 6 different teams. Most important, of course, are the biological readout people, because without that, there's no use to any of this. So.
Thank you very much, Chandra. Very, very interesting talk. 2 questions. One is about the stapling of the peptides. So, are there any rules in exclusion criteria for stapling?
Okay. Yes. Very good. And second question?
Yeah, that's about P53. So I'll come to the break.
Huh?
That's about P53. So I'll come to that.
Okay. So now the question, what he's asking is the following question, which I skipped that slide. In fact, I removed that slide. But the question really is here. Remember I said that you have to predetermine. And I say it very pompously as though we know the logic. We have no clue, to be honest. Essentially what happens is you carefully scrutinize this. Let's say you have a complex and you scrutinize this and you ask yourself, by introducing the staple, which are the interactions I'm likely to disrupt that make the peptide bind to it? So far, this question as to, how far apart should these two be? This is II plus 4, which means that it spans the first residue of a helix and the fourth residue. We have examples of II plus 3, we have examples of II plus 5, II plus 11. We have examples of II plus 4 and II plus 5, both. There's a variation in how they work. It varies across systems. So the simple answer is there are no rules yet. It's an open field, for someone to establish rules for how and where to stabilize these. Why is that important is because some smart guy one day will come around and develop a biological system to staple things. Part of it has already been done with the group of one of Tom's colleagues called David Spring we work with in Cambridge. And they have already begun to develop this method of, trying to have these things happen inside the cell. See, right now we are doing this outside the cell. So you're unable to do anything automatically as a screen. So, but the other thing that this technology, Pavan, helps is this one, the one there. So if you look at this technique, this will already provide you hints. If you look at this, so this density that you see here is looking at all the staple peptide crystal structures and asking whether this method can reproduce all the regions where the staple should lie. So if you use a technique like that with your peptide, then it's already giving you hints of where the staple should lie. And you can see very nicely. It captures all the densities. And this wasn't biased by the structure. This was, the superstructure is superposed later to see. So this is another method that you can add. After looking carefully, looking at the rules that people like Srinivas and all develop, then you add this aspect to it because the thing is these are polycarbons. That itself makes them a little bit less amenable to rigorous rules so easily, as we know for amino acid changes. Amino acids, as rightly pointed out. But maybe someone will discover these hot spots of the kind that have been talked about yesterday and now he's mentioning. So.
Thank you. The second point was about the P53-MDM2 interaction. Now, as an outsider to the field who doesn't understand this whole area, I was thinking, just as a very So I was thinking, is it possible to, let's say, install, like we have this genome editing field coming up very rapidly for the last 3 years. Now, is it possible to design P53 molecule in such a way that you eliminate this binding pocket and then you install this gene right in the genome itself, and so you churn out P53 molecules that do not have the binding affinity to
Right. So, if the story was that simple. No, you're right. It's a very, very good point, because, the problem is that the next step in this process is assembling the transcription machinery. Okay, so I don't have beautiful slides like Tom was showing, but the next step is it binds to P300. Okay, and paradoxically, the very same recognition motives that enable it to bind MDM2 and get alleviated as a result of this are also enabling it to necessarily bind to P300. And if it doesn't bind to P300, there's no transcription. So you might as well have a non-effective P53. Yeah. So that's why I'm saying it's So what the effect of this is on the next step of P300 is hardly been explored as well. Yeah. So you may ask at this point, hey, you're designing these peptides, they might go and bind to P300. Of course they do. At least in the computer we can see that they do. The extent to which, so here is a matter of numbers and the matter of residence times that we don't know anything about in any field, right? Is that how long do you need to inhibit MDM2 for P53 to come here? If that same molecule inhibits P300 a bit weakly in the competition, is it sufficient that you just require a little bit of activation in those cells? Those cells are so stressed, right, with these oncogenic stresses, that maybe you just need a little perturbation in the right direction and the cell will die. Yeah? And maybe that is why it is working, because these staple peptides for sure are going to inhibit P300 as well. But we just think that from our models that they are not doing it so effectively, and that it allows sufficient activation of a binding of P53 by itself to the remainder P300 molecules that enables the activation. Yeah? Srini, you have another question?
If there's no further questions, I will ask.
Ask.
So, regarding the dynamics of density, what will be the simulation if it is done with an amino acid that contains molecules?
Yes, so now the field is wide open. So there are people who are developing this. So when Tom talked yesterday about fragment-based discovery, using, the much more functional assays they have developed in Aztecs, there are a lot of people who are developing computational techniques of trying the same thing in the computer, where you have not so much your amino acid, but the functional groups that are required to guide the design of your molecules. So imagine you have an asparagine with an oxygen carbonyl here and an NH2 here. So now you may have a small carbon atom with CH2 and a carbon with a carbonyl and an NH2 here, CH, or you may just have a carbonyl and an NH2, and you can saturate your system with anything. Yeah, there's no restriction on this. You can make it as artificial as you want. That's the beauty of computers. You could do all kinds of stupid, nightmarish things. At the end, you need a testable hypothesis. That's the critical thing. We do whatever we do. At the end, if we do not have an experiment that, my Pavan will do successfully, then it is all a waste. I mean, it's nice fun to keep us in jobs, but really waste. So,
Just 5 more minutes.
Yeah, I was looking at the watch. I thought we were not very yet. Okay. My understanding, to be complete, is that the role of state is, or rather, one of the roles of state is to preserve the helical nature.
yes.
I just wanted to have your views on using an amino acid like AIB. Not
Yes.
in lieu of.
No.
statement,
Yes,
but
absolutely.
in addition to statement.
Absolutely.
And you also seem to want hydrophobic amino acids.
Yes. Yes, you're absolutely right. And
you know, when it is used in association with L amino acids,
yeah,
it will form the right-handed helix. It can have only two conformations.
right.
Either the right-handed
Right.
helix or
Right.
left-handed
Right.
helix.
Right. Yes, very good point. And so AIB is a precursor to this. So for every system, we first test it with AIB as well. Now there are two issues happening with AIB, as there are many issues with this. So in this, the amino acid I showed you had this polycarbon moiety here on one of the carbon instead of the hydrogen, the other has a methyl. Everybody thought that, yeah, this is needed to maintain that rigidity. There's this guy in Leeds called, I forget his name. And he says to me, no, I don't need it. I can just have a hydrogen there and it works as well. Point one. Second point is that a lesser number of AIB containing molecules are going inside cells. Somehow the staple is helping you get inside cells. Yeah. In the AIB case, that for some reason is not so far possible. Yeah, you can't rule out, you know how biology is. Anything that cannot happen will happen. So I think that, you know, you are very, very right. But those experiments are always done in tandem with the staples, just to make sure that, you know, there is not a great degree of change in affinity. The affinity increase that you get from a staple also comes from that interaction with the surface in a lot of cases. But having said that, oh, it's gone. If you look at an HIV protease that developed a staple inhibitor peptide, there it works very well, but the staple points out into solvent. Okay, so nobody is yet bothered to see whether the effect of that hydrophobic moiety into solvent is actually a good thing or not. We don't understand these things, right? Because we don't understand the nature of things such as what is the role and interaction of water molecules with these hydrophobic regions and what kind of pressures they create for a molecule to nicely sit in or not. Yeah, it's a field that's slowly evolving, but it's a very complex field to try and understand the specific interactions between water. And I think that comes back to this question, because I didn't talk about this, that when we just looked at this simple interaction in terms of water dynamics, you find very, very interesting differences that you can actually in the end manipulate to make better peptides, that we have done in one particular case, but it's just a one case. And I think we just got lucky more than anything else. So, yeah, but it's a very interesting and very valid thing that anybody who's working in this area should also use AIB amino acids, absolutely. Because it gets rid of that nightmare of that reaction. Yeah, okay.